Okay. Last Thursday we saw a what I think is a wonderful film depicting Watson and Crick and Franklin, and I must say that era of history, for those of you that are students of history, and if you're interested in science and history, that whole area of Wilkins and Franklin and Watson and Crick has now been written about. Those players have been, except for Franklin, who died shortly after the discovery of the double helix, have been interviewed. It's fascinating history, the whole relationship between Watson and Crick and Wilkins and Franklin, and perceptions in the film, which was taken from Watson's book, and it was Watson's perceptions. All of those things have been written about and discussed by colleagues who were at Cambridge at that particular time and other times, and it's amazing. So if any of you are interested in like an honors project, you're interested in writing about that era, thinking about it, it really is terrific if you're interested in the history of science. One of the interesting things about that period was that Franklin is portrayed in a particular way. And the way in which he was portrayed in the film and the book has been the object of an enormous amount of debate and research over the last 50 years. Be that as it may, as I said, Franklin died tragically and prematurely of ovarian cancer about 7 or 8 years after she worked on the DNA double helix. But it should be told, despite what you might read in the book and what might you have seen on the screen, in her last days of living, she lived with Crick. And it was Crick and his wife that actually took care of her. And she became absolutely very, very good friends with Watson, Crick, and Wilkins and all of these players in the time after the discovery of the DNA double helix. So let's just play a little game before we move on and answer one question and have a little bit of a discussion, not for very long. Keep in mind that a Nobel Prize is given for seminal discoveries in a variety of different areas. And the Nobel Prize for the discovery of the DNA double helix was won by Wilkins, won by Crick, and Watson. And it's only given to 3 people. 3 people, and that's it. It can't be given to any more than 3 people by Nobel Prize rules. And it can't be given if a person is not alive. So it can't be given posthumously. And so we'll pretend that you're on the Nobel Prize committee, and you have the choice of choosing any of these individuals for winning the Nobel Prize for discovering the DNA double helix. And so let's take a shot of this and see exactly what you would do. A, Watson, B, Crick, C, Wilkins, D, Franklin, E, Gosling, F, Chargaff. Go. Going once, you got it. Stop. Where's Brandon? Thank you. There we go. So, 29% of you said Watson, 18% Crick, 11% Wilkins, 43% Franklin, very interesting. And no one said Gosling or maybe Chargaff. And since there's only ability for those choices, that's essentially what we have. Okay, so those are interesting answers. So let's just debate those for a second. Okay, so most of you haven't finished the book, but I want you to. Because as I said on Thursday, double helix is the greatest biological discovery of the 20th century, and perhaps the greatest in the last 100 to 200 to thousands of years. Can you just turn it up a little bit, and I'll save my voice. Thank you very much. So, who said that Watson should share? Which of those of you on that committee, thank you, that's perfect, said Watson should share in the Nobel Prize? And I want you to think critically here and give me some very specific answers. So who said Watson? Zach, we got a microphone? Why don't we pass it down? Thank you. It's off.
It was kind of a. It was between Watson and Crick.
No,
for
but
me,
just give me a straight answer.
I thought
don't
he
need
was
to
the
have a
one who was
dissertation.
pushing it the hardest. He was the one who people were interested in it, but he was the one who was really pushing them to investigate
But what did
it.
Watson determine that was very unique to the structure of DNA?
The helix shape?
No. What did Watson come up with? But you put Watson on your Nobel Prize. You're going to vote. You voted for him, correct? Okay. What did Watson determine? Brian?
Watson put the backbone on the outside and then the pairing of the bases on the inside.
That's okay, but what did he really do? Eden?
That A bonds with T and C bonds with G?
Yeah, he got the base pairing rules right, which is absolutely central to the double helix. Okay, how about in Davis? Who's the second person in your Nobel Prize? Faria?
I said Crick just because he's also he worked closely with
what did Crick
Watson.
do, though? I mean, he worked closely, but I might have worked closely with him, too, if I lived back then. But that doesn't mean that I would get the Nobel Prize. So the point is, what did Crick do that was unique to solving that amazingly simple but incredibly difficult structure to solve?
I'm not sure if wasn't he the one who figured out the shape, the helix shape, or?
Well, you're on the right track. Does anyone have anything to add about down here about what Crick did? Let's hear from somebody else. Samantha.
Okay, so when he was looking at the x-rays that Franklin had taken, he was able to realize that the
And
shadows...
why was he able to realize that? Why was Crick able to do that?
Because of Franklin?
No, it had nothing to do with Franklin. Why was Crick able to recognize what those X-ray diffraction pictures meant? Do you remember from the film?
Did it have to do with the research?
Lauren?
Didn't he
How's your
develop
parents,
a theory
Lauren?
for? Oh, good.
Good.
Didn't he develop a theory for reading?
Yeah,
X-ray
exactly. He was the
crystallography?
one that developed the entire theory for translating the X-ray diffraction data into structural data. And remember he said, oh, that's, group C2. And he said, remember in the film, group C2? And he said, it goes this way and it goes this way. I mean, it was like a dyad. And so that was absolutely critical for being able to put the helix together. So we have Watson and we have Crick. And let's see, who's the third person? Who put Franklin on their list? Davis? Who has Franklin on their list? Dora, do you? I'm just speculating.
Yes.
Okay, tell me why.
Because she did most of the research and took most of the photographs.
Mm-hmm. So she did most of the hard research.
Right.
And she had those beautiful photographs, right?
Right.
So now we sort of have a chicken and an egg problem, don't we?
Right.
Why do you think we have a chicken and an egg problem, Dora?
Because although she took the photos, she couldn't figure out.
Yeah, exactly. So we got a little bit of a problem now. She took the photos. She's crunching that A structure with this Patterson transformation, which is a mathematical transformation of the X-ray data into structural data. And you saw that little machine, which I used to use way back when, I think probably sometime in my life. But the point is we probably could do it on a PC now in about 10 minutes. But the fact is, tell me about the chicken and the egg, Dora.
So she took the photos of the double helix.
Yeah.
And but she couldn't figure out what she discovered. Like she couldn't tell you that it was a double helix. She just had a photograph.
So then how come you put her on the Nobel Prize?
Because if she didn't take the photos, then no one would have.
But a lot of people win Nobel Prizes for data that other people generate, but they don't have the idea. There was just a Nobel Prize awarded for the people that discovered the AIDS virus. But the guy that discovered that virus actually caused AIDS, Robert Gallo, wasn't even given the Nobel Prize. In fact, there was an article in this week's Science magazine signed by about 500 people saying that the Nobel Committee really blew it, because if it wasn't for his work, no one would have known that the AIDS virus caused AIDS. So do you want to still, I'm not trying to influence your vote, by the way. Who else has Franklin? Rose. How many poems do you think have been written about roses throughout
Lots
all
and
the
lots.
time? Huh?
Lots
You
and
had
lots.
to guess.
That-
Millions, hundreds, thousands.
I'm going to say
What's your
millions.
favorite Rose poem?
The one that comes to mind is not a poem, but from Romeo and Juliet. So I don't.
And go ahead.
A rose by any other name. I don't know.
Go ahead. So how come you have Franklin?
For me, Franklin, she, despite the fact that she didn't actually come up with the structure with like the bases and whatnot the way Watson and Crick did, it was from her report that Crick saw like the C2 structure or whatever it was in the movie. That's how they portrayed it, that Crick saw that she had figured out it was C2, and then he was able to translate that information from her X-ray using his theories into actual structural
Okay.
data.
We can argue either way on that. I don't have a problem. It was a different time in science, a different time in culture. Who said Wilkins? Who put Wilkins on your Maxime? Max.
I put Wilkins
You
because
didn't. That's like you're at the dentist or something, you know?
both my parents are dentists.
Go ahead.
so-
They're dentists? That's why.
All right. Wilkins already had.
Speak up.
Wilkins had work before.
Can you hear us at Davis?
Yes.
Great.
He was the first one to kind of recognize that DNA probably had the gene in it, and so he was.
Positive about that?
I'm guessing, but
Yeah, you're guessing. You're such a bullshitter.
yeah.
Go ahead. That's not true.
Oh, okay.
In any case, you've got Wilkins there. Give me some specific answers.
Well, he was already guessing that it was a helix before anyone else, and he was asking for Rosalind to give him the X-rays, and even though maybe if he would have got
Well,
them,
let's
he would
not,
have guessed.
I mean, let's be very specific about this.
All right.
You have Wilkins, you awarded Wilkins a Nobel Prize for what reason and what reason only? I'm not interested in how he related to Franklin or how he didn't. What was his contribution to the elucidation of the double helix? That's the bottom line. What is it?
His just the backing of the structure that was found by Watson and Crick.
Who else has Wilkins on the list? Chris. I need a microphone. And I need a very specific answer, not, oh, I believe, or I think, or I wish, or I hope.
I was just going to say that I
Sit
kind of.
up. We're not, in a spy here.
I think I might want to second guess that answer. I just raised my hand because there's only, like 3 people. But I couldn't really think of any specific reason why I would have awarded him. It may have existed, but I don't remember why, so.
Anyone else put Wilkins on their list? So we have Watson, Crick, and Franklin. Okay, different question, but I'm just going to phrase it for you. I don't have it typed. Who was the individual among these 6 individuals that had the original idea that the DNA had a regular structure and that you could solve its structure by using X-ray diffraction methods? Who had that idea to study the structure of DNA using X-ray diffraction before anybody else had that idea? Was it A, Watson, B, Crick, C, Wilkins, D, Franklin, E, Gosling, F, Shargaff? So it's only one. You're going to vote for just one person. A, B, C, D, E, or F. Go. Stop. Wilkins. So do you mean to tell me that the person who had the idea, that the person who had the insight, that the person who proposed that you could actually study the structure of a molecule using X-ray diffraction and the structure of DNA and had the first data that suggested maybe that it was a helix wouldn't share in the Nobel Prize? long before any of those people came into that game? Remember, Watson knew Wilkins was interested in the structure of DNA and the X-ray diffraction. Franklin was then hired after Wilkins started his work, primarily because she was really the world's expert, or let's say one of them, at doing that kind of stuff. But Wilkins was the person that had that idea. And so I think, and I'm just speculating, I don't know what would have happened if Franklin had been alive. Again, it was a different time, different history, different culture. Lot of gender discrimination, unfortunately, during that particular time in history. I'm not quite sure what would have happened. It's a hypothetical question. But certainly the person that had the original idea to do this and stimulated all the work certainly deserves something in terms of sharing, in my opinion, that particular prize. Okay, let's move on. In order to understand genetic engineering, of course, we got to understand what a gene is. And we have to understand what the gene is, what it does, how it works, how it controls things in the cell, how one manipulates it, and all the really goody things about genes, which I love talking about, and I'm going to share some of my insights with you. And so for today, what I'm going to do, is I'm going to talk about genes and gene functions, but I'm going to go back in time again and talk about some very important experiments leading to the discovery of DNA as the genetic material, tell you a little bit about that time, and then I'm going to give you my bird's eye view on what a gene is all about. And following that, what we're going to do is we're going to have you look at some DNA, which I can see is running on this gel right up front. So you're actually looking at a gel electrophoresis experiment going on. And then you're going to have a little bacterial cloning experiment towards the end of class. So let's talk about genes and DNA. So remember, we live in the age of DNA. And remember I said last week that every single thing is possible. We can synthesize DNA, we can clone DNA, we can take one gene from one organism, put it in another organism, get it to work. We can basically do anything. It's absolutely unlimited. Okay, so in order to really understand this thing called genetic engineering, of course, we've got to be able to understand what a gene is, what it does, how they work, and everything else. So let's talk about that. The first thing is, and I think all of you know this, that genes specify functions inside cells. There's absolutely no question about that. We have a collection of genes, and now there's some new words here, which is called the genotype. So all the genes in your genome, the whole collection, the sequence of the genome, all of the genes in your genome is called the genotype. And those genes specify your collection of traits as you look around the room. That is your phenotype. And that's because the genes then specify that particular genotype. And so genes really exist for only a couple of functions. Genes exist for replication. That is, they have to perpetuate themselves absolutely identically from generation to generation. But of course, they don't always do that, as you'll see. So that's the first important function of genes. And then secondly, they have to be able to transmit some information to the cell to tell the cell what to do, to make hair color, to make skin cells, to make heart cells, to make whatever it is, eye color, whatever it is. It's all dictated by genes. And so in a sense, as I said, the genotype is the collection of genes. They have to be replicated, cell to cell to cell to cell, exactly the same. Those genes have to then tell the cell what it is. That is, they have to have some sort of activity to specify the traits in the cell. And then finally, we have the functions. All of the functions in the cell, either directly or indirectly, are the results of gene action. Could be direct gene action or indirect gene action. What do I mean by that? Direct gene action would be that the gene specifies a protein, and the protein, let's say, is in your hair and specifies the color of your hair. That's direct action. Indirect action is, stay awake, Eric. Indirect action is the gene specifies a protein. It's an enzyme. An enzyme is responsible for a process, for example, like DNA replication or some other process in the cell. And therefore, the final result of that process is because of a chemical reaction. The gene's not involved in that chemical reaction. The product of the gene, which is the enzyme, is involved in catalyzing that reaction. That's sort of more indirect. But the point here is that genes specify all cellular functions. Again, replication, gene action, cell function, and proteins. And this is extremely important. And so, as I said last time, genetic engineering, and I'm going to say this again, is no hocus pocus, no BS, no beliefs, no spirits. You're just intervening in this process, DNA to RNA to protein. When you genetically engineer a cell, you're inserting a piece of DNA into the chromosome of that cell. It replicates with that cell just like all the other genes in that cell. It's expressed in that cell just like all the other genes are expressed in that cell. And ultimately, if you add a new gene, like a glow gene to a plant, you get a new function, you get a new trait, and you can visualize it. The important thing to consider is that when you look, as I said, at outward traits, it's very important for all of you to keep in mind that it's the result of the action of genes. In other words, if we manipulate traits by breeding, and I showed you last week how all the different vegetables were engineered in one shape or form by our ancestors, or the animals, the domesticated animals, like cats or horses or goats or sheep or whatever. All of these traits that our ancestors engineered for, behind them is the action of genes. And so what's absolutely critical to understand from a genetic engineering point of view is if you engineer a trait by classical breeding, you're manipulating, in a sense, this molecular process even though you're not visualizing it. When you engineer a trait using modern genetic engineering, you're intervening directly in that process because you know the trait that you want to alter, you have the gene, you can add it to the genome, you can get it to be active whenever you want it to be active, and you can do whatever you want with it. So what I'm saying here is, and this is extremely important, is that when you look at these traits, you know, Mendel's traits of round and wrinkled and yellow and green, what you see is the action of genes. And what you're seeing here is simply when you do breeding, manipulating genes outwardly, when you're doing, let's say, molecular genetic engineering, you're actually changing the DNA, you know, because you're changing the genes that you want to alter. And so we have replication. Any of these green or yellow peas can be perpetuated generation after generation. And we then have, let's say, the green gene making a green protein, giving rise to the green seed. Or we might have a yellow gene giving rise to the yellow seed. Harumi, tell me why, again, we have purple cabbage and green cabbage. Remember? Or we have green seeds and yellow seeds. What's your hypothesis?
It's due to mutations.
It's due to mutation. So this is important. So all genes replicate from generation to generation, and most of the time that replication is absolutely perfect. Maybe 1 out of a million or 1 out of 10 million times there'll be a mistake. And that mistake leads to different forms of the same gene. In other words, the gene might have a particular sequence of nucleotides, which we'll talk about today, and you may change one nucleotide pair in that sequence, and that generates a slightly different RNA and a slightly different protein. It's still the same gene. So for example, round and wrinkled, that's a trait in peas due to one gene, but wrinkled is due to the mutant form of round. Yellow and green are due to the same gene that produces the same color, but yellow, you can assume maybe, is the mutant or the alternate form of green. The point here is that genetic variability is generated in a population. And again, look around the room. We look different because of genetic variability. We all have the same genes here or in Davis, but we're different because those genes take on different forms on a population level. And any given gene can have an infinite number of different forms. There's no limit, for example, to how many different forms a gene can have. And that genetic variability is very important. And it was that genetic variability that our ancestors used to make broccoli, cauliflower, to make big tomatoes from small tomatoes, to make corn from its wild-type ancestor. And these things arise spontaneously in that replication process by these rare mutations. Now, I said last week that there's a very important word. Remember what I said, Dora, that word was allele. It's a very important thing that we're going to be talking about throughout the entire quarter. An allele is simply the different form of the same gene. And that different form differs because the sequence of that gene may differ between the alleles by one nucleotide, or two nucleotides, or three, but not very many. And so different forms of the same gene lead to allele, and alleles lead to genetic variability, and genetic variability leads to us. Genetic variability leads to all of the vegetables we eat in the grocery store. Genetic variability leads to all the animals, Chihuahuas, Great Danes, all the things that I said. But it's just the process of gene activity and due to the functions of genes. And genetic engineering creates new genetic variability, as we'll talk about. So let's go back to the tomatoes that I talked about last week. And you can see all different forms, shapes, sizes, colors. All of these things occur because of mutations in genes that specify these traits, rare mutations, giving alternate forms which then can be used and were used to select for those things by breeders. And then notice that these genes are on chromosomes. And notice that the different forms of the same gene reside at the same place on a chromosome. And this is very important conceptually because of the fact that it's the same gene. So we have, let's say, yellow tomatoes and red tomatoes. They reside at the same position of a chromosome. And in fact, as you'll see, the same position of a DNA molecule. They're not going to change position because it's the same gene, just an alternative form. Julie, do you get that? Anna, do you get it? So we look different not simply because you're female and I'm male, but we look different because we have different forms of the same gene. You know, that's very important to consider. And it's extremely important because if we didn't have different forms of the same gene, we wouldn't be around and no other creatures would be around on the face of this earth. What genetic engineering does, from a molecular point of view, is the ability to create this genetic variability. And as I said last week, we can do this from scratch. We can create any gene, synthesize any gene, put any genes together in any combination, make anything we want, do anything we want in a laboratory. We can put any gene of any organism in another organism, and there's absolutely no limit. And so the point here is that that's what genetic engineering is all about. And so now, Dora, can you give me a hypothesis for how the glowfish experiment shows that DNA is the genetic material? And why don't you have Faria help you? Because both of you can do it together. So what is your collective hypothesis for how the glowfish experiment that we talked about last week, in fact, shows that DNA is the genetic material? What do you think?
Because all organisms share similar DNA.
But how does that experiment show that DNA is the genetic material? How does that experiment demonstrate directly, let's say, that, the function of genes? How does that experiment demonstrate absolutely that if you take some naked DNA and you do something using genetic engineering, that you'll get something else? How does that experiment demonstrate that DNA is the genetic material? What do you think? What's your hypothesis? Faria, do you want to help?
I think because of the fact that we all share the same genetic
Do you think or do you know?
Well, it's my hypothesis, maybe. I'm not sure if it's right, but the
There
fact
is
that
no right
we
or
all,
wrong hypothesis. Go ahead.
because the fact that we all share the same genetic makeup, that we can insert genes that can possibly show in other creatures.
Okay, so then how does that experiment demonstrate that the DNA that's taken out of the jellyfish that specifies the fluorescent protein is in fact the genetic material, using this process up on the screen as an example?
The fact that it could actually work in the fish
When you
shows
say
that.
work, what does that mean? Put it in molecular
Express.
terms.
Like, yeah, look, expressed.
What's-
The trait was actually expressed.
It's expressed. How do you know that it's expressed? What's the evidence for that?
Fish glow.
It glows. The fish glows. So you put a piece of DNA in, it's expressed. That's correct. So it glows. That's what you see. How do you know that the DNA is able to replicate itself? What's the evidence for that? What's your hypothesis, Faria? Remember we said that genes have to replicate themselves and have to tell the cell to do a specific thing. What do you think?
Because, well, the fish around it, are they just, did it insert
Well,
each fish or?
What do you think that the, let's get someone down here to help. Derek, here you go. Why don't you talk to Dora and Faria up here. Can you do that? Can you turn around and, can you see him? There you go. He's this guy right over here. He's being zoomed in on. Get his head there. Now you can see him. Okay, talk to those guys. Not me. Talk. Can you see Derek up there?
Yeah.
He's a biochem major, a junior biochem major. He's following in your legacy, John. Okay, so how does this experiment show that DNA is the genetic material? One, Faria said because it's able to, you're putting the DNA, let's say, in a fish from a jellyfish, and you get this fluorescent protein. But how does it also show that the DNA is being replicated?
You see the expression.
Talk louder, talk up. I know you're a quiet guy.
We're able to see all of the fluorescent proteins being expressed in all of the cells. So
Okay, good. That's a really good thing. So if you put it in the egg, and then you see it active in all the cells of the fish, it had to replicate as the cells of the fish divide. So that's absolutely excellent. But what else? Is there only one fish now that has that particular protein?
the trait should be able to be passed by
The parent?
Yeah, the parent.
Yeah, so it should be passed on from generation to generation, and it is. So in doing that experiment, one can demonstrate, in fact, that DNA is, in fact, the genetic material. So then we come back and we summarize here the properties of a gene. So if you were working in a dark room with blinders on and sunglasses over your blinders, and you knew nothing, remember Watson and Crick discovered the DNA double helix knowing nothing except some scattered facts. And what's true genius is to be able to make something from unconnected facts, and that's what they did. And so let's say you're working in a room and you know nothing about genes. You don't even know that a gene exists. You don't know what their substance is. You don't know that they're DNA. You don't know that they're protein. You don't know that they're RNA. You know nothing about genes. But you want to determine whether some molecule in the cell is the genetic material. It has to have certain properties. Does everyone agree with that? Absolutely has to have certain properties. And those properties lead to predictions. And so we have the scientific method again, which is what I talked about last week. So what are the properties? Well, we've just discussed them. First property is it has to be able to replicate. You have to be able to transmit something from generation to generation. That means whatever molecule is the genetic material has to be able to replicate generation after generation after generation after generation. Anna, do you think that process of replication is simple or complicated? Simple. It looks simple, but the nitty-gritty, is it complicated or simple? Yeah, very complicated. But we have to be able to replicate. So that means the molecule has got to have some mechanism Not simply replicating, though, but passing a blueprint for a phenotype on for generation to generation. That's the key. I mean, it could replicate and not do anything. But this is passing the blueprint on to say every time the skin cell divides, the skin cell is in fact a skin cell. Or every time the liver cell divides, it's a liver cell. Or every time we produce new human beings. That's the essence of that molecule which carries those genetic instructions from cell generation to cell generation. And then secondly, this molecule has to be a little bit stable. Can you imagine, Ben, if we had an unstable molecule, which is our genetic material, we'd never be here, would we? Red?
No, we wouldn't.
Yeah, so the point is that it's stable, but it can't be completely stable.
Yes, because otherwise organisms wouldn't evolve.
We wouldn't be here either. And so it's got to be stable, but not completely stable. So it's got to be stable, and then also it's got to be, would you say, pretty universal?
Yeah, all organisms should have it if they all...
They have to have something that it may not be the same substance. It turned out that it was. But they have to have something that specifies the phenotype and the traits and stuff like that. Would you agree with that?
Yes.
And then finally, it has to be able to tell the cell to do something. And so the question is, how do you carry out some experiments in order to show that? Well, we conducted experiments last week in class that demonstrated DNA was a genetic material. We took a bacterial toxin gene and we stuck it in a plant and made it insect resistant. We took the glowfish gene and stuck it in flies and in rhesus monkeys and in mice and in plants and showed it was a genetic material. We took a growth hormone gene from a rat and stuck it in a mouse and made a big rat and showed that that naked DNA, meaning purified DNA, was the genetic material. We took the male-forming gene from a human and we stuck it in a female mouse egg and it became a male. All of those experiments show that with one piece of DNA, purified, no protein, no anything else, that piece of DNA can specify a trait and can be perpetuated from generation to generation and therefore is the genetic material. What do you think, Marvin? Pretty exciting stuff, huh? You think they knew about that in 1918? Can you talk into the microphone so we can hear you?
No.
What do you think?
I don't think they knew about it in 1918.
Maura, what do you think? You think they knew about genes in 1918? What year do you think Mendel discovered the laws of genetics? 1732, 1956, 1432, or 1865? What do you think?
Those are all really good options.
What do you think?
I don't know.
Well, let's take a vote. We don't have the eye clicker, but let's take a vote. How many people say that Mendel's laws of genetics were discovered in 1450, around the time Columbus discovered America? Yes or no?
No.
How many people say that Mendel's laws of genetics were discovered in 1750, sort of during the Renaissance time? Yes or no? Derek, you say yes? You're looking around, you know. If everyone in the room voted for McCain, would you vote for McCain? If everyone in the room voted for Obama, would you vote for Obama? What do you think, yes or no? No. When was Mendel's laws of genetics, Kathy, discovered? When do you think? 5 years ago?
No.
When? Have the microphone.
1865?
Yeah, 1865. So Mendel did his work in the middle of the 19th century. Unfortunately, the monks in his monastery didn't like what he was doing because he talked about sex, and they burned all of his papers. That's true. And Mendel's laws were undiscovered for another 50 years. So we know about the laws of genetics in our time basically because of the discoveries of Karl Korns and Alexander Schermak, a German and a Russian, who rediscovered independently Mendel's laws about 1905. And the word gene was invented in about 1910. And so the word gene hasn't even been around for 100 years. Isn't that pretty amazing? So we knew nothing about the laws of genetics 100 years ago. It's absolutely amazing. I said that, I think, last week, and every time I think about that, especially because there was this woman who's 114 years old, the world's oldest person that they highlighted in the Los Angeles Times this week, and she was born before the time that we even knew there was genetics and the laws of heredity, and she's still alive today, and we can look at her genome and look at all the different genes and say, how come she can live so long? I think that that's absolutely amazing. So let's go back in time. Let's go back about 80 years or so. Faria, what was the major cause of death in 1918? Major cause. It's not the one on the board. What do you think? And don't look ahead because that's cheating.
Not really sure. A flu, maybe, or something.
No, it's not the flu, but flu was a big killer. How about diphtheria? Whooping cough? They were the major killers, things that we have antibiotics for now that hardly anyone gets. Although I must say, in this new age that we live in, there are many parents today that won't vaccinate their children. Can you believe that? Even though their child could die from whooping coughs, could die from diphtheria, could die from measles, like that, and yet won't vaccinate for reasons that maybe we'll talk about later on in the quarter. So from 1918 to 1920, as you can see on the board, approximately 20 to 100 million people, 1/3 of the population of Europe. This was a pandemic called the Spanish flu. Can you imagine how many people were killed? It's absolutely astonishing. Due to influenza, which is caused by the flu virus, which is still around. And furthermore, we now know what the exact gene is that was responsible for the flu pandemic of 1918. How do we know that? Well, Dora, we know that because people dug up corpses in the Arctic permafrost, dug them up not too long ago, took tissue from those corpses, found the virus, the flu virus, cloned the genome of that virus, sequenced the genome of that virus, of course under very severe, P5 containment conditions because you don't want to let that thing out again. And then they mutated every single solitary one of those genes using genetic engineering. And in a report just published this month in the Proceedings of the National Academy of Sciences, said this is the gene that produces the protein on the flu virus that's responsible for the 1918 pandemic. Isn't that absolutely amazing? Using genetic engineering to find the cause of a disease that killed 20 to 100 million people, which hasn't been seen, thank God, on the face of this earth over the last 80 to 90 years. The point here is that when you go back in this time period of 1918, 1919, 1920, there's massive death around you. Everyone has someone who's dying because of an infectious disease. Cancer in those days was not the major killer. Heart disease was not the major killer. Why? Because people died when they were 15, 16, 17, 18, 19, 20. They never got old enough to have heart disease or to have cancer. And so imagine if you were a scientist or a physician at that particular time in history, you would also look around and say that the flu was also a major killer. Influenza, either primarily bacterial in those days. And influenza was a major killer because, one, it was opportunistic. If someone came down with the flu, respiratory disease, pneumonia also occurred, and it decreased their chance of survival. And I must say that the survival rate for the Spanish flu was 1 in 2. Half of the people that got it died. Imagine that. This was a major killer, bigger than AIDS, bigger than anything we've seen since then. And the flu, as you can see by this thing that I took off of the New York Times. And by the way, for those of you who don't know this, You can go back in the New York Times archives all the way back to 1865 and read anything that's been printed in the New York Times since that particular time period. So the key here is that we have major killers at this particular time in history, bacteria and flu, and even today we have 1 million deaths due to pneumonia every day. That's why you should wash your hands. There is the flu bacteria, Streptococcus pneumoniae. You can see it. The blue is the cells of your lungs. The red is the bacteria. There are the major causes of death in 1920 compared to 2002. If you were living in this particular period, typhoid fever, malaria, smallpox, measles, scarlet fever, whooping cough, diphtheria, flu is way down here, mumps, cholera. Every single solitary one of these diseases is treatable either by vaccine or by antibiotics. We hardly even see any of these things, except, as I said, you might see some measles because parents don't vaccinate their children. You might see some kids tragically dying due to typhus or whooping coughs or diphtheria because they don't get the shots that you guys have all had, because we all get these booster shots and things like that. But all of these are treatable, curable diseases. Now, look here. Heart disease, infectious and parasitic disease, this is all over the world combined. So you can imagine all the different kinds of diseases that cause that. But cancer, stroke, AIDS, digestive diseases, very few of them are bacterial and viral diseases except for ones that are lumped together. And so that's the miracle of science. That's the miracle of antibiotics. When were antibiotics first used, Eden? Can I have a microphone? I need microphones.
Here, I got one.
There you go. Thank you.
When penicillin
What
was
year?
discovered,
When were they first used? In large amounts? In 1903? In 1852? When do you think?
1903.
No. When do you think? Take another guess. Nancy, when do you think antibiotics first came into massive use? It's surprising, actually.
I don't know, like the smallpox vaccine was
We're talking
like
about antibiotics, not vaccines. What do you think?
antibiotics. I think the Egyptians did something with
I don't
Rodenburg
think so.
penicillin.
I don't think so.
No,
I don't think so.
1850s,
What do you
I
think?
guess.
What year? Give me a year.
1850s?
Faria, give me a year. We have 1850, 1830, antibiotics, 1900. What do you think?
1870s.
How about World War II, about 1941, as recently as that? Remember, you can discover the antibiotic, but you have to mass produce it. And that's sort of the topic of discussion this week. You can clone a gene, but if you want to make a product, you have to mass produce it. And so antibiotics are not that old in terms of their use. So imagine before the era of antibiotics. Okay. So let's tell the story of DNA as the genetic material. I love this story. It's one of the most remarkable biological stories around. So let's go back to 1918, 1915, 1920, and imagine there's deaths all around you. Everyone is seeing someone die due to pneumonia, due to tuberculosis, due to the flu. No family escapes this. If you're a physician living in this particular era, all you're doing is seeing people die, and you're trying to just make them comfortable. There's absolutely no way to prevent it. There's no vaccines. There was somewhat of an understanding of what the causative nature of the disease was, but there's really nothing more we can do about it. So enter a guy by the name of Frederick Griffith, who lived about that time and who was a microbiologist physician, and who had his lifelong goal to be able to make a vaccine. That's what Griffith wanted to do, to be able to make a vaccine that could prevent the flu, influenza, due to this bacteria, Streptomyces pneumococcus. And so Griffith really was a remarkable guy because everything that we do with genetic engineering, every single solitary thing that I showed you last week, traces its origins to the experiments of Griffith, although he never understood what a gene was, He didn't know what DNA was. He didn't know what genetic engineering was. He really knew very little about the molecular basis of what he was doing, but he did some experiments that proved to be absolutely essential in this march towards understanding DNA as the genetic material. He invented the word transformation for reasons that you'll see in a second. So what did he do and what was he working with? If you look on the board, you see two plates. Red means that these bacteria are growing on a plate in what's called blood agar. Blood, sort of a media of blood, which provides all the substances for the bacteria that the bacteria needs to grow. We have another one which is growing on just agar supplemented with carbon, hydrogen, nitrogens, and again sugars and vitamins and all the things that's necessary to grow. Now I know you can't see it, but these are called smooth and these are called rough. And Griffith was working with these two forms of the pneumonia bacteria. And as I'll show you in a second, Griffith knew that the smooth form was virulent, meaning if you took the smooth form of the bacteria and you injected it into a mouse, the mouse died. It's a very important fact. And he knew that spontaneous mutants of smooth, in other words, smooth could mutate into rough. They could distinguish the morphology of these cells by looking at them on a culture plate. That these rough cells, which are spontaneous mutants of these smooth cells, were avirulent. You inject them into a mouse, and the mouse lives. Now, Eric, wouldn't you imagine if you were living at that time and everyone was dying, and you had two forms of the bacteria, a virulent form and an avirulent form, you'd want to know what was responsible for causing the avirulent form? Definitely. Because if you understood what caused the avirulent form, you may be able to understand the basis of the disease. If you understand the basis of the disease, you might be able to affect either a vaccine or a cure. Particularly a vaccine. If you knew the molecule in the bacteria that was responsible for the disease, you might be able to do something to prevent the disease, even in the primitive land that they lived back then. Smooth and rough forms can be considered allelic variants of each other. It's a mutation, let's say, in the same bacterial morphology gene. Smooth can mutate to rough, and rough can mutate to smooth, although very rare, maybe 1 out of 1,000,000 times. But you can get different forms of these same cells, and you can distinguish them by looking at them with a microscope and seeing what their different structures were. Okay, let's talk about bacterial cells, including the pneumonia cell. First of all, keep in mind that bacterial cells are single cells. They're prokaryotes. They live by themselves, generally. They have one large circular chromosome, generally attached to the inner membrane of the cell. And that's depicted right over here. They also have, remember it's a circle, they also have plasmids. And as you know, those plasmids carry antibiotic resistance genes. And it's those antibiotic resistance genes that prevent the bacteria from being killed by these viruses, in a sense, or not viruses, by fungi that produce the antibiotics. But they also have, my head was getting ahead of me, they have restriction enzymes also that prevent the bacterial virus from killing it. So we have a single-celled entity, a large circular chromosome with many, many, many thousands of genes, small extra chromosomal circular DNA capable of replicating by itself that have antibiotic resistance genes that combat antibiotics that are trying to kill the bacteria. We have restriction enzymes in the cytoplasm that fight off foreign DNA like viral DNA, as we've talked about, in order to combat lethal viral infections. And then we have a cell wall, which protects it, made of carbohydrates. And under the cell wall, we have a cell membrane made of fats, meaning lipids, in order to transport things in and out of the cell. Because imagine it's a single-celled entity. It's living in a sewer someplace. It's got to take things from the sewer, get them in, and use it for growth and all kinds of different stuff. It's a very sophisticated cell. It's about 2 to 4 microns in length and about 1 to 2 microns across. A micron is 10 to the minus 6 meters. 10 to the minus 6 meters. It's very small, 3.94 times 10 to the minus 5 inches. And so those are the dimensions. A typical bacteria circular chromosome has anywhere from, as you can see, 2,000, 500 to 5,000 genes. It's a lot of genes. The smallest one is 500. And I must say that, as I showed you last week, although you didn't make the connection, we can use now synthetic biology to synthesize from scratch. Using genetic engineering, we can, in fact, synthesize the genome of the smallest bacteria and insert it in bacterial cells and get the bacterial cells to live. So we now have the capability, as of last month's article, which was just published, of being able to synthesize an entire genome, stick it in a bacterial cell, and have that cell live. So we have a genome which has been synthesized from scratch, which causes the bacteria to live. Plasmids are much smaller. They have 1 from 100 genes. And so they're pretty nifty creatures. And keep in mind that these bacteria are the results of billions of years of evolution. They're highly sophisticated organisms. And they do things in a very sophisticated way. And here's the variety of these things. And here's a conceptualized version of what those bacteria look like. But more importantly, their genomes have been sequenced by genome projects. And what you can see here is that they have many thousands of genes. Here's hundreds of genes to thousands. And this is the bacterial genome that's been sequenced from this Mycoplasmid genitalium. It's a bacteria that infects, it's a venereal disease, essentially, in humans. And this has it's the smallest genome that exists on the face of this earth of a unicellular organism. And that's the one that's been sequenced and put back into cells and shown that those cells can survive. So we have bacterial genome projects now, which provide a lot of information about what those bacteria do and what those genes do. That's the Streptomyces. This is the Streptomyces pneumoniae bacteria. And this particular bacteria has something which is novel to many bacteria of this kind. It has a cell membrane that's used for transport, as you can see. It has a cell wall, but it has this magic capsule, which is called a sugar capsule. And this is the capsule that makes the smooth bacteria. This capsule makes the bacteria look smooth on the plate. And so if we look at the plate with the smooth bacteria, they're smooth and they glisten because of that capsule. And when smooth mutates to rough, that capsule is gone. When smooth mutates to rough, the capsule is gone. Dora, can you give me a hypothesis for why the smooth form of the bacteria is virulent and the rough form is avirulent in mice? What's your hypothesis? Remember, I just told you that the smooth form has the capsule, the avirulent form does not. What's your hypothesis? You want some help?
Yes.
Thomas, where's the mic? What's your hypothesis? We take the smooth form, we stick it in a mouse, the mouse dies. We take the rough form, we stick it in a mouse, and the mouse lives. What's your hypothesis for the difference?
The antibodies wouldn't be able to penetrate the capsule.
What's your hypothesis?
I hypothesize that the antibodies can't penetrate the capsule.
That's exactly right. So the antibodies from the mouse can't do anything because the capsule protects it. Without the capsule, the antibodies destroy the bacterial cell, and that's the difference between virulent and avirulent pneumonia bacteria. That's absolutely correct. Okay, so let's go to the experiment. We have a circular DNA in the genome. That's sort of obvious, and there we go. Okay, Griffith. Griffith noticed that when we have a smooth form, as I said, it can mutate into a rough form, and it can go back. We have the rough form, which is avirulent, and the smooth form, which was virulent. And he's working in 1926, 1925, 1928. And what Griffith wanted to know was, how can I make a vaccine against this horrible killer bacteria so that I can, do quite a bit to prevent people from dying from this scourge, which it was at that particular time. So he did an experiment, which you can read about. It's published in the Journal of Hygiene Research. It's a British journal. He published it in 1928. You can get it online. You can read exactly what he did. But there's no logic to his experiment. In other words, Max, Griffith really didn't know anything about genes. Griffith didn't know about DNA. Griffith wasn't interested in gene action. Griffith wasn't interested in DNA. All he was interested in was trying to find out what was going on with rough and smooth and virulent and avirulent. So he did a lot of empirical experiments. An empirical experiment is you sort of just do something. You're not really looking for the mechanism of something. You're just doing something in order to get some observations. And so he did the following experiment, which turned out to be one of the luckiest experiments that anyone has done in the history of biology. And so, like all experiments, experiments have controls and experiments have, tests. And so, first of all, he took the smooth form of the bacteria and he injected it in a mouse, and the mouse died, as you can see. That's the control. Smooth form, virulent, put it in a mouse, replicates in the mouse croaks, deader than a doornail. You look inside the heart of the mouse and you find the smooth form of the bacteria. But then what he did is he took the rough form of the bacteria, simplistically. The experiment was actually a lot more complicated than this. And he injected it in the mouse. And this was a mutant variant of the same smooth form that he had already used to kill the mouse. Nothing happened. The mouse lived. And furthermore, when he looked in the mouse, there was absolutely no bacteria because, as Thomas said, the immune system destroyed the bacteria. So far, so good. That's okay. Just give me another one. Yeah, thanks. So far, so good. Okay. So now, he was trying to understand how you could get rid of this bacteria. So he boiled the smooth form of the bacteria. He boiled it. Just cooked the heck out of it. And he took the heat-killed bacteria, and he injected it into the mouse, and of course the mouse is going to live because the bacteria is dead. Now here was the most confusing experiment. And it was confusing to Griffith because he didn't really understand the result. And listen carefully. He took the live form of the rough bacteria, in other words, the rough avirulent bacteria, and he mixed with that live form of the rough bacteria deader than a doornail, heat-killed, smooth bacteria. Get it? He took live rough bacteria. They lived. They weren't virulent, but they lived. He boiled the smooth form and killed it. Nothing survived because he had the control over here. He mixed them together, injected it in the mouse, and the mouse died. Brian, would you have predicted that? What do you think? Why not? From the controls, would you have predicted that result?
No, I wouldn't have.
Tell me why.
Because when we inserted the rough, they didn't die.
Speak up.
When we inserted the rough, they didn't die.
When you injected the rough, you didn't insert it. When you injected, the mice didn't die. And what about?
And when I injected the heat-killed smooth, they also didn't
die. But now you put them together and the mouse died.
That's true.
What would you have done if you were doing that experiment? What would have been the first thing that you would have thought about? What would have been the first explanation you would have thought about?
That they must have transformed.
I don't think you would have thought about that.
Oh, at the time?
I mean, you might have thought about that fast forward, but what would you have thought about if you did the experiment? Catherine.
Oh, I guess I'd be pretty confused.
Can you sit up? But what would you have thought about with your little, what was that, Cub Scout shirt or Girl Scout?
Cub Scouts.
Yeah, okay. What would you have thought about? What would be the first thing that comes to your head?
Maybe like.
What's the first thing? No maybes, no. What's the first thing comes to your head? See this result? Mouse died. How can that happen when I have these results here?
the dead cells somehow like infected or
They're dead.
or changed.
They're
I know.
dead.
But they could have shared
And how
DNA.
come you didn't see anything over here? You have dead cells. How come they didn't do anything?
Because they didn't have any like. Well, I would have thought they just would have shared DNA like the.
They don't know anything about. You got to go back in time, Catherine.
Then I would
no
know.
knowledge
Then
of
I would-
DNA.
I would have
no
no
knowledge
idea.
of gene processes. Get yourself out of 2008. What do you think, Kathy? On.
I would think that I did something wrong.
Ah, say that louder.
I would think that I did something wrong.
That's what John says to Ryan all the time. Ryan, you must have done something wrong. Do the experiment over again. Isn't that correct?
Hey. No.
No? Yeah, that's absolutely right.
I'm usually the one who thinks that I did it wrong.
Yeah, so maybe you did something wrong, or maybe some contamination was there. So you do it over again and you get the same result. And you do it over again and you get the same result. And you do it over again with different isolates and different times, different places, and you get the same results. And it becomes very, very, very conclusive. And so they didn't know what the transforming principle was, but it was a beautiful experiment.